It is a pleasure for me to introduce the first speakers of today, Dr. Lieve Herman. She is the head of unit on technology and food at ILVO in Belgium. And Lieve has a very long-standing experience in the risk assessment of microorganisms and microbial products in EFSA. So, Lieve, the floor is yours.
Thank you, Pier Sandro. It's a pleasure for me to be here in the Parma Summer School with you and to present to you an introduction on the risk assessment and how EFSA is handling this for microorganisms. A diversity of microbes are used in the agri-food chain. There are viruses, bacteria, yeast, fungi, microalgae. And these microbes can be used as a natural isolate, the wild type strain, but they can also be conventionally mutagenized. They can be genetically modified. And at the end of the spectrum, they can be really synthetic microorganisms. A synthetic microorganism is based on a minimization of the microbe. So the microbe is only minimized to its essential functions. We call that the chassis, like for a car. And then several metabolic blocks are introduced in this chassis to form just a dedicated microorganism to produce certain compounds. So the risk assessment at EFSA spans all these kind of microorganisms from non-GMM, non-genetically modified microorganisms, still till this synthetic microorganisms. There is also an increase in the agro-food chain of the introduction of microorganisms and their products. And this is mainly due to the reduction in costs for producing these microorganisms and their products. So in the past, it was more expensive, and then it was more dedicated, this microbial production, towards more products with a higher value, like in the pharmaceutical or the cosmetic sector. But now we are more and more evolving to a cellular agriculture where microorganisms are taking an important place, more and more important, and we are expecting that this will also increase in the future. An example is the production of stevia. Stevia, which is normally produced by a plant and which is now produced by a yeast in which the whole metabolic pathway for the production of this stevia from the plant has been introduced in this yeast, so that this yeast can now produce this stevia product. When a microbe or its product has to be introduced in the market, this has to be risk assessed by EFSA. And this is a requirement due to legislation for a lot of these products. So products introduced on the market, produced by microorganisms, can be feed additives, enzymes, vitamins, amino acids, here also the animal probiotics, plant protection products. More and more important, the use of microorganisms as plant protection products. That's because more and more the chemical products are banned to be used, and therefore there is an aim to look for microbial solutions. Also enzymes, food additives, flavorings for food, food additives, biomasses, so the real microorganisms themselves, so the biomass or the single cell proteins introduced in the food chain as novel food. Also when there is a health claim, they come to EFSA, and also when a genetically modified organism is introduced, these enter the EFSA risk assessment. An important remark is that starter cultures used in dairy production or in meat fermentation are not coming to EFSA. They are not the regulated product requiring an EFSA risk assessment, and also not the human probiotics. So the animal probiotics, they are coming and they are risk assessed by EFSA, but the human probiotics, they are not, and also not the biostimulants for plants. The risk assessment at EFSA starts with an application dossier by the applicant. So the applicant, so an industry who wants to put something on the market, has to prepare a dossier. And this dossier, this requirement, is stipulated in the different regulations which are in place in Europe for the different products which can come to the market and which can be produced by microorganisms. There is the regulation related to the genetically modified food and feed, to feed additives, to foods with health claims, food enzymes, additives, flavorings, novel foods, and the plant protection products. You see a whole battery of legislations. And to guide the applicant, you find on the EFSA website the information to make up your dossier. And you see here, depending on the legislation or on the area where your product or your strain belongs, you can find the necessary information for making your dossier. Here you see the flow of applications. So the dossier is introduced at the European Commission, which make a suitability check. It comes to EFSA. Also there will be a suitability check. And when the dossier is accepted, it entered the EFSA procedure for the risk assessment. The risk assessment leads as a results in an opinion which will be adopted and discussed by the respective panel, and then it will be published in the EFSA journal. When now you go somewhat deeper in the risk assessment process of microorganism, an important basic concept is the categorization of the microorganisms and their products in 4 categories. And in fact, for practical use, it's reduced to 3 main groups. So group 1 contains the category 1 and 2 products, and that are the purified products. It are products resulting from fermentation, and these products are purified so that no microorganisms are present and also no DNA. So that are the category 1 and 2 products, and examples are enzymes and amino acids. And frequently these are produced by genetically modified microorganisms. A second group are the category 3 products. In these products, the microorganisms are killed, so they are inactivated, no living or no viable microorganisms still present, but their DNA is still there. Important examples are the biomasses. Biomasses which are now increasingly coming to the novel food for market organization and which become a substantial part of our food and feed production for sure in the future. An example which is already on the market is corn, which is produced by Filamentous fungus, Fusarium venenatum, and which was on the market before 1997, so that it was not considered by the novel food legislation and has not undergone this EFSA risk assessment, which is now in place for the new biomasses which will enter our food column. The third group is then the category 4 products. That are products where you have intact living microorganisms introduced in the food chain. An example or important examples are these plant protection products. For example, the spores of Bacillus thuringiensis, which are used as plant protection product and which is a category 4 product. For all the categories of products, so category 1 till 4, molecular microbial risk assessment is in place, also a toxicological assessment, the assessment of the allergenicity, and also the technical purity related to the manufacturing process is considered. For biomasses, so the category 3 product, which become or can become a substantial part of our consumption pattern, also a nutritional assessment is required because it's clear that these food ingredients need to be nutritional valid to be introduced in our consumption. For the category 3 and 4 products, because there are viable organisms and our DNA present, also an environmental risk assessment needs to be carried out. For example, for plant protection products for category 4, it's important to check if how far these microbes are also acting towards non-target microorganism in nature. And also for category 3 product, the possibility of the horizontal transfer of DNA needs to be considered. For example, when the microbe would contain an antimicrobial resistant gene, it's important to consider that this antimicrobial resistance could eventually be transferred to other microorganisms and be of concern later on. So I said already that all category products need to undergo microbial molecular risk assessment. And this is in EFSA based on the whole genome sequence of these microorganisms. This is at the moment mandatory for bacteria and yeast, still optional for fungi, and not yet ready for microalgae, just because of technical reasons to have this whole genome sequence on a routine based to be carried out. But it's, you see, this is an evolution which will not stop, and this whole genome sequence analysis is forming the basis. It's used for the species identification, also for the characterization of the genetic modification, for the search for antimicrobial resistant genes, and for genes of toxins and virulence. For category 1, 2, and 3 products, also the absence of viable organisms need to be documented. Like I said before, it's the basic of this categorization and of the fact how far an environmental risk assessment is necessary. And for category 1 and 2 products, the absence of DNA from the production organism need also to be documented. EFSA is also providing in a statement. I give the links at the end of my presentation so that you can check the details if you would like to do. But in this statement, detailed requirements are given for carrying out this whole genome sequence and to give an idea what is acceptable during our risk assessment. So in this statement, you will find how the identification of the microorganism based on the sequence data can be done. There is a necessity to compare it with the type strain of this taxonomic unit of this species, and can be based on digital deoxyhybridization or the average nucleotide identity. And also the thresholds are provided in this EFSA statement. Also, you find information how you have to document the genetic modification, with, of course, the necessity to describe the insertions, the deletions, and the substitutions made in the genome of the microorganism. And also for the identification of the genes of potential concern, also here guidance is given for the search of the databases for antimicrobial resistance. It's recommended to test at least 2 databases, and also for genes of concern because of possible toxigenicity and pathogenicity. In whole, the risk assessment, the QPS concept can be of a big importance. QPS can provide a fast track assessment, a fast track through all this procedure. QPS stands for Qualified Presumption of Safety, and it provides a safety status for microorganisms which are used in the food and feed chain. Important is that this safety status is provided at the species level. And this means that it covers all the individual strains which can belong to a certain species. And all these strains are then covered and can profit from this fast track assessment. You find the list of these taxonomic units, so these species for bacteria, yeast, and fungi, and fungi not, but microalgae. You find this list on with this link on the EFSA Knowledge Junction in Zenodo. Also, genetically modified microorganism can use the QPS concept. In the case, the recipient strain, the basic strain, has qualified for the QPS status, and when, of course, the genetic modification would not introduce a concern. For each application arriving at EFSA, you remember the flow, the application is coming to EFSA. There is the suitability check at the app desk, at the application desk. When the application desk discovered that in this dossier a microorganism is involved, then they will notify this dossier he will notify this dossier to the QPS working group. So these dossiers, these microorganisms, these notifications, they arrive at the QPS working group, who start then to evaluate the QPS status of this organism. So they do an extensive safety evaluation of the species towards the strain of the dossier belong. And when the QPS working group consider this species, this taxonomic unit, valuable for QPS, then they will publish it, and each 6 months the biohazard panel adopts a statement in which these new QPS notifications are evaluated. And when they are positively evaluated, this microorganism will then be incorporated in the QPS list. So the QPS list is constantly updating with new microorganisms. It's also constantly updated based on the available literature. So each 6 months, in the same statement, which is adopted by the biohazard panel, all the existing taxonomic units on the QPS list are again evaluated. So each 6 months, the QPS working group is evaluating about 6,000 literature papers related to the QPS taxonomic units to evaluate if no new concern would have been reported and if the QPS status can be kept for all the organisms on the list. Just to draw your attention also on the notification list. So there are 2 lists. There is the QPS list, but there is also the notification list. And this can be interesting for you because the notification list, which is also publicly available, can give you an indication, give you information on all the dossiers arriving at EFSA. So all the dossiers which are have an involvement of a microorganism, you can check them on these notification lists. So these notification lists include QPS and non-QPS organisms, so they have an overall impression of what organisms are notified in dossiers to EFSA. Just a small summary of the QPS list. At the moment, it includes 106 taxonomic units, 62 species of gram-positive non-sporulating species, mainly lactic acid bacteria, 18 species of gram-positive sporulating bacterial species, mainly bacillus species, 3 gram-negative species, 17 yeasts, 3 virus families, which are used as plant protection products, and 4 protist microalgae species. So many people don't know that also already 4 microalgae are already on the QPS list. There are some species which are excluded from the QPS assessment, and that's because some strains of these species are known to contain, for example, for fungi, Omycetes and Streptomycetes, genetic information to produce mycotoxins or secondary metabolites or antimicrobial products to be produced, or then for some other species like for Enterococcus escherichia coli, some virulence of toxigenic potential of some of these strains. When you look to the QPS list, you will find there also the qualifications. So it's an Excel table, and you will find some columns indicating qualifications. What is a qualification? In the case that scientific knowledge identifies a specific hazard which is only related to some strains of a taxonomic unit and which can be easily assessed at the strain level, then we introduce a qualification. And we say, okay, this hazard has to be excluded at the strain level during the assessment of the dossier. Just to give you some examples that will clarify it better, I think. For all bacterial taxonomic units, there is a generic qualification. So all these bacterial QPS bacterial taxonomic unit, the absence of antimicrobial resistant genes have always to be confirmed at the strain level. So that's a qualification. Because some Bacillus species, some Bacillus QPS species have strains can have toxigenic potential. Also, that has to be confirmed. The absence of it has also to be confirmed at the strain level and is a qualification for these Bacillus species. For a lot of QPS organisms, you will also see as qualification only for production purposes. So this means that the end use of this microbial organism, which is covered by QPS, is restricted to the production process. This means that is restricted to those applications where no viable cells are present. So not for category 4 products, only for 1, 2, and 3 products. So it's okay for the biomasses. And also for some yeasts, which are used as viable cells, for example, as animal probiotic, then they are used as viable cells. There, the absence of antimycotic resistance has also to be confirmed. Like I said already, these qualifications need to be assessed at the strain level by the respective areas at EFSA who are assessing the dossier. Just to remember that also for dossiers where a QPS organism is involved, the dossier needs still to contain some information. It's not that you don't have to add nothing in your dossier related to the microbe. First of all, you have to identify your organism, just that it's clearly confirmed that it is indeed a QPS organism. And of course, you need the qualifications to be set. You need also to indicate the safety of your manufacturing process and the safety of your genetic modification. But you don't need to extensively have this toxigenic assessment, which is otherwise required for a non-QPS microorganism. A very frequent asked question is, how can I get my microorganism on the QPS list? And I understand this question because a lot of people would like, before they start an innovation or before they start a concept or a new idea, to know if their microbe could become on the QPS list. In 2007, when the QPS concept was born, when it was introduced, then there was a public consultation. And then people and industry could ask, notify their microorganism to EFSA. And that was independent of an application dossier at that time. But from 2008 till now, the only way to get your organism on the QPS list is through a notification to EFSA. As a consequence, and that's very important, the QPS list is not exhaustive. So there are organisms which have the potential to come on the QPS list, which are safe, but which are not yet on the QPS list. I've introduced the two last slides of my presentation on the GRAS concept of the United States, just to make a comparison with the QPS. And that's because it's very often, it gives a confusion very often, and both terms are sometimes mixed, and they are really different. In the United States, when you want to get your product on the market, you have a new food additive or a new food ingredient, you want to introduce it on the market, you can do it by just sending a dossier to the FDA. It's a bit like you would do it in Europe, but you can also do it based on the GRAS status, the generally recognized as safe status. But in this case, the company is introducing this status for their peculiar product they want to introduce. And then the FDA can evaluate this status and of course can also challenge it. Also here I give you some links so that you can get more information and also consult the list of these GRAS status products you can find for the US. So here I made a comparison between GRAS and QPS with the main differences. GRAS, like I said, is product related. It's the company who gives this or introduce this GRAS status for this product. That's different from the QPS because the QPS is independent of the product and independent of the production process. The manufacturing process is evaluated separately from the QPS assessment. So the GRAS is therefore related to a specific product or production organism. The QPS is general. It includes all the strains belonging to a certain taxonomic unit. So it's much more broader in the strains it includes. The GRAS status is determined by external expert companies. It's only reviewed by FDA. So the literature searches are done by the companies, while the QPS is totally in hands of the of EFSA. The EFSA experts are doing it and are also continuously updating it. Like I said, each 6 months we continuously do all the literature searches and update this QPS list. You see here the companies are asking for the GRAS status, and here it's in fact it's only through a dossier that the QPS assessment will be introduced to EFSA. I have included in my presentation some slides with a lot of interesting links where you can find all the details of what I've now summarized in this presentation. So the most important guidance documents I have here included. There is the EFSA SAP Panel 2021, the scientific guidance for the submission of food enzymes. This is a recent guidance with the guidance also to the molecular and microbial assessment of these microorganisms and in the frame of the food enzymes. There is also the guidance for the feed additives from 2018. The EFSA statement, which I introduced already, on the details of the whole genome sequences. You have the link here as well. And the one on the genetically modified microorganisms, which is still in place for category 4 and 3 and 4 products which are genetically modified. Here, the last slide with the links to the QPS information, information on the topics, the link to the QPS list, the link to the notification list, and also the further information on QPS.
3 minutes, Lina.
I think I've here come to the end, Pier Sandro, of my presentation, and I want to thank all of you for your attention.